I thank UHS Bagalkot and the organizers of the National Symposium on Chili for providing me with this opportunity. I'll try to finish as quick as possible. Can I have the slide? I'll not get into the details. Only thing that we need to look here is, of the 5 species that we have, Capsicum baccatum is the only source for resistance to anthracnose, as we know. Apart from baccatum, we also have chinense, which is resistant to, which carries genes for resistance to I'll not get into the productivity and okay. 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 ಇದನ್ನು ಹೆಂಗಾದ್ರೂ ಸರಿ ಸರ್. Yes, sir. Okay. Coming to the dry chili production, we have Telangana, Andhra Pradesh, Karnataka, and Madhya Pradesh as the major contributors to dry chili. And keeping this in view, we have framed certain work. Of the several biotic constraints, we have anthracnose as one of the major biotic constraints. And to understand this, we first try to look into the resources for anthracnose, that is coltotrichum. These are the prerequisites for resistant breeding: understanding the disease-causing pathogen, identifying stable sources of resistance, knowledge on the genetic basis, and DNA markers linked to genomic regions controlling resistance. The first thing is to understand the pathogen. Anthracnose, as we all know, is caused by a complex group of coltotrichum species, of which Coltotrichum gliosporidis and capsaicii are very predominant in India. Now they are called as truncatum and siamens. And Coltotrichum, capsaicii, and gliosporidis contribute to 50% of the yield losses. And coming to the resources for resistance, baccatum and chinense are the only 2 sources of resistance that we have. Okay. Now we carried out a survey of the 3 major states, Andhra, Telangana, and Karnataka. We collected about 130 infected samples. We characterized them, and these are the places, and we characterized them using different protocols. We looked into the concentrations of molecular characterization, DNA marker approach, and then we looked into the colony characters like the colony morphology. And then we use species-specific primers to detect them, and we also use AFLP markers to characterize these fungal isolates. And both the phenotype-based and the molecular characterization reveal that there are 2 species in India, that is Coltotrichum capsaicii and Coltotrichum gliosporidis. Then we screened one of our hybrids, a private company hybrid, with all these isolates, and we identified 2 isolates, CC39 and CG33, sampled from Khammam district of Telangana, to be very, very virulent, and we have been maintaining these isolates and using them in our routine screening programs. Then the next step was to screen our accessions with these virulent strains of Coltotrichum, and we identified contrast in the form of Susan Joy, which expressed a lesion size of 30.4 mm and PBC 80 with a very, very low lesion size. Similarly, for gliosporidis, we identified PBC 80 as resistant. These are some of our in-house lines that we found that they are These are the right side panel shows the in-house lines that were resistant to anthracnose. These are the cultivated types that were resistant. And here you could again see PBC 80 and 81 are the resistant sources. These are the resistant sources. Then as a first approach, we made crosses between PBC 80 and annum. And we used CA14 that had 29 millimeters. And what we found was there were reciprocal cross differences in terms of the seed formation itself. When we used PBC 80 as the female parent, there was no seed set. So we had to interchange, and then we found annum as the female parent and Bacatum as the male parent produce seeds. However, all the seeds that were set were aborted. So the next step was to rescue the embryos. But that took a lot of time, and we now have a protocol wherein we have been able to recover an F1 hybrid embryo between Gauri Bidnor, a local line. Today morning they were mentioning that Gauri Bidnor is not available now, but we have Gauri Bidnor seeds with us. Gauri Bidnor into PBC 80, and Gauri Bidnor also happens to be one of our parental lines of the hybrid. One hybrid that we released, the parental line is Gauri Bidnor. We have sufficient quantities of those seeds. Here we have now recovered a plant that is now at that stage. At the right panel you see that we have the F1 hybrid embryo between Gauri Bidnor and PBC 80 recovered. However, we started with a second approach. We wanted to understand the genetics of anthracnose resistance. Then we involved two Bacatum lines, a resistant Bacatum and a susceptible Bacatum. SB is susceptible Bacatum. These are the plant types and the fruit characters. And the response of the Bacatum susceptible and resistant lines are as follows to Coltotrichum capsaicei and Coltotrichum gliosporidis. And we made a cross, we derived the F1, we sell the F1 to derive the F2, and the F2 was screened for the anthracnose disease. And we classified the responses into immune, highly resistant, moderately resistant, and susceptible. And later on, you'll see that the first two classes, the immune and highly resistant, are categorized as resistant, moderately susceptible, and highly susceptible are classified as susceptible. And we used a special scale to record the lesion size so that we could convert a qualitative character into a quantitative character. Because this is the experimental setup in-house. And this is the distribution of the overall lesion diameter. And the lesion size ranged from 1 millimeter to 11 millimeters in the C. bacatum, intra-C. bacatum cross. And then we tried to identify a marker that was linked to it, and we found SSR marker E081 to be linked to resistance. However, we were not able or successful in transferring this marker into annum. Then we started a third approach. We made a cross between two annum lines that expressed moderate resistance. And they were Lampang local and Gauri Bidnor. We derived a rill population. And this is how, through single seed approach, we developed the rill population. We tried to map the trait. And then the lesion size ranged from 7 to 30 millimeters in the F2 and also in the rills. And what we observed was, again, the process is the same. We identified lines that were resistant to anthracnose. About 8% of our rills were found to be resistant. And we tried to tag this locus. And these are the recombinant inbred lines that are promising for fruit yield as well. We found HPMS E047, an SSR marker linked to the trait. However, the amount of variation it explained was very, very low. Again, we failed. Breeding for anthracnose resistance has been a failure, but we are now progressing towards some results. This is the response of Susan's Joy. In the first couple of slides I showed, Susan Joy is a contrasting parent with a very, very high lesion size. We use Susan Joy as the female parent crossed it to PBC80. And we derived the F2. So this is the shape of the F2. And then we also backcrossed it. And when we backcrossed it, this is the advanced backcross derived line SP1. Susan Joy into PBC80. And this line has shown tremendous improvement in terms of the response to anthracnose infection with lesion sizes less than 5 millimeters. However, the fruit shape and the fruit characters are not very encouraging. This is the fruit shape. Not very encouraging. And then the next thing that we did was, having this in mind, we did a Jiva study, including 180 germplasm lines, and then we found, using 250 markers, of which 166 were polymorphic. Then we found these genotypes, PBC80, 81, SP1, and other lines reporting to be resistant to anthracnose. These are the resistant lines. All of them are Bacatum. And this result is from Jiva study. Then we found some markers associated, 3 markers associated with, putatively associated with anthracnose resistance, that is collotrichum infection. And these 3 markers were now used in our further studies. For this, we generated another cross. Based on some studies, we studied both Mendelian inheritance as well as the quantitative trait inheritance. And we wanted to know the number and mode of action of genes controlling resistance. So for that, we made a cross between S343, a highly resistant genotype to LCV earlier, but now resistance is broken down, with the SP1, an in-house developed line. And we developed the F2 and also the backcrosses. We used the 6 generations to understand the genetics of the trait. And for this, we have the assistance from Namdari seeds as well. One of our doctoral students is working with the Namdari seeds, and we are progressing towards some results. Well, this is where we are studying, and well, we classified the 5 classes, the plants, F2 plants, into 2 groups, resistant and susceptible to understand Mendelian genetics, and we found that the traits segregate in the ratio of 1:15. 1 resistant is to 15 susceptible, so indicating that it is a duplicate dominant epistasis for susceptibility, while homozygous recessive condition is resistant. The good part is the resistance is fixed in the F2 itself. So this can be forwarded directly. But the problem is, for every 16 plants, we will be able to recover only 1 resistant plant. So to recover 25 resistant plants, we have to get at least 400 plants. And out of the 400 plants, we'll have 25. Then 25 plants will not carry all the desirable or horticulture traits. So we need to raise a very large population to identify desirable plants. And this is being done both at GKVK and Vidadi, and this is the results of the Mendelian inheritance pattern. Okay. So this is anthracnose disease resistance is controlled by 2 recessive genes with duplicate epistasis in S343 into SP1 background. Now, the breeding implications is, I told you, we need a large population to get hold of resistance as well as horticulture traits. Then we had identified some markers in the Jiva study, and we used these markers to tag the locus. And what we observed was, yeah, 2 markers, CAMS 451 and GPMS 185, are found to be associated with resistance, explaining a phenotypic variation up to 25%. So this is the stage where we are. So we have markers linked to resistance. Then we try to find out these are the markers and their sequences. Markers HPMS 016451 are on chromosome 1, chromosome 5, and chromosome 8. However, they are distant from that of the locus controlling anthracnose distance. This is where we are. And this is the F2 population that we have derived from S343 into SP1. The sad part is all the fruits have a blunt tip, the character of S343. So we are now looking for a plant that has desirable fruit morphology. So,
Rao?
Yes, sir. I'm done, sir. I'm done. Now, a host of PhD students work on this. I thank all my PhD students for their continuous efforts. I thank UHS Bangalore because chili is not a mandate crop of UHS Bangalore. Despite that, they allowed me to work on the crop for the last 15 years. I thank Kirkos Trust UK. Professor Sir Edwin Southern funded our project for some time. DBT New Delhi also funded our project. And Namdari Seeds we are collaborating with. Thank you. Thank you for your attention.
At the end, we are not where we need to be, right?
Yes.
People don't accept failures. So I congratulate you for that. Because it takes time. You have done an excellent work. But there is my 2 bits worth of suggestion. Number 1, when you are doing interspecific crossing, please take the wild type as male. You will get better at your crossing success. Number 2, don't take the direct F2 if you want a good type. You must do 2 or 3 back crosses. That's why you are getting the blunt one. Please take 2 or 3 back crosses to your recipient parent or wherever you want that, so that you can increase your better genotypes. For academics, you did what you were absolutely right. That is how you will know. You can make RILs today with DH. You could have made RILs if you have that facility. Then you can do in 1 year all that F2, etc. need not be made.
Yes, sir.
So those are the two things I want to add. And you also brought out a very good point. We don't understand that we need a very large population if we have to be successful. My friend in Israel, an erstwhile colleague in Pioneer, is Yanni Yelkind, a very legendary capsicum breeder. He, in our webinar once, he said, I am doing 10,000 F2 plants, and that to BC1 F2. So please keep this as my suggestion. There is no question. Excellent work. Thank you.
Thank you, sir. Thank you for the suggestion. To inform the House, we have 10,000 F2 seeds of this crosser.
Which species of anthracnose you have used for market development?
For market development, Prototrichum capsaicii.
Anthracnose, but which species?
Prototrichum capsaicii and Gluosporidis, madam.
That SP1 is annum or baccatum?
SP1 is a derived line from baccatum again.
Crossed with?
PBC80. We cross Susan's Joy with PBC80, and back crosses we derived SP1.
Do you think Susan's Joy is capsicum annum only?
Yes, sir.
Susan's Joy.
Susan's Joy is annum.
It is cross-compatible with baccatum?
Madam, with very great difficulty, we got only 3 seeds of this cross, of which 1 plant we retained, and from that we progressed further.
Sir, in our work we used Chinans as a bridge. We could get some success.
Yes, madam.
Naresh will be presenting later.
Yes, madam. One of our students, Dr. Ganesh Prasad, who is now working for a seed company, is here in this hall. He did the bridge cross as well. Annum into Chinans and then into baccatum.
Is that marker getting validated in baccatum also? The population what you developed, CB into CB?
No, madam, we didn't do that. We didn't do that now.
Thanks.
This is a recent one, 2024-25. One of our PhD students is working in collaboration with Namdari Seeds.
I think we are little
We
ahead.
have shared the population with them.
Our work is little ahead, I feel. Naresh will be presenting that.
Okay, madam.
Thank you, sir.
Okay, madam.
Thank you, Dr. Rao. I think, sorry, because one more request has come with the additional paper. I don't know whether we'll be able to manage time. Quick question. Last question.
Good evening, sir. Myself, Mr. Sharan from Indo-American Hybrid Seeds. Sir, you mentioned that PBC 80 is tolerant to Gliosporidis and Capsicum.
Yes.
So, does it confirm resistance to Sovile and Acodatum also? Because of late, Anthracnose is appearing on the green fruit as well, sir. And there are reports this green fruit Anthracnose is caused by Coleotrichum Sovile.
Sir, we did not test this. We did not test this, but we'll have to do that. We have not yet tested, sir. Acutatum, we did not test.
Sovile, sir, Sovile.
Yes, sir.
Thank you.